T-cell acute lymphoblastic leukemia, also known as T-ALL, is an aggressive type of leukemia that affects children and adolescents. It progresses quickly and affects the T lymphocytes, which are a type of white blood cell that assists B lymphocytes in making antibodies that fight infection in cancer cells. Over 50% of cases of T-ALL have a mutation in the Notch1 gene, making it a popular target for treatment. Existing treatment involves gamma secretase inhibitors that target the Notch1 pathway. Inhibition of gamma secretase prevents the Notch intracellular domain from being released from the membrane, preventing the activation of Notch target gene expression. However, these inhibitors are inefficient in causing apoptosis in T-ALL cells and have been found to have negative side effects in the gastrointestinal system because the inhibitors are nonspecific, affecting both cells with and without the Notch1 mutation. The Notch1 gene is first transcribed and translated into the ER. Once processed in the ER, it is transported to the Golgi apparatus. At the trans-Golgi, a furin-like protease processes it to produce a heterodimer with two subunits, the extracellular subunit NEC and transmembrane subunit NTM. The heterodimer is then translocated to the membrane, where the NEC interacts with the DSL. The interaction between the NEC and DSL initiates further cleavage of the NTM subunit, the last of which is performed by gamma secretase. The final cleavage results in Notch intracellular domain, NICD, a transcription factor that then translocates to the nucleus to activate genes involved in cell proliferation. Cassiaria sylvestris is a herb found naturally in Brazil that is used to treat inflammation. A compound from this herb, Cassiarin J, is a tricyclic claridinoid that directly inhibits the vital calcium pump in the sarcoendoplasmic reticulum known as the SERCA pump. The SERCA pump transports calcium ions from the cytoplasm to the SR. CJ is preferentially cytotoxic to leukemia cells. The reason why and the mechanism is not known. Our main case study examines research performed by D. Ford et al., which examines the effect of CJ treatment on T-ALL cells. In this study, it was found that the treatment of CJ with excess calcium in media caused the intracellular concentration of calcium to increase. The calcium storage in the ER was then emptied into the cytosol. IP3R is a calcium channel located in the ER membrane. Inhibition of the SERCA pump by CJ directly caused the IP3R channels to release the calcium within the ER into the cytosol. To buffer some of the excess cytosol calcium, the mitochondria take some in, which results in oxidative stress, which was measured with the ROS. An increased ROS is associated with CJ treatment, which leads to oxidative stress that causes activation of proteins involved in the apoptosis pathway. These experiments were performed on various T-ALL cell lines, but CJ's effects were strongest on those that had an HD domain mutation on Notch1. NFKB is a transcription factor important in the regulation of the survival and growth of T cells, and the activation of this pathway requires ER stress and increased intracellular calcium. An active Notch1 activates NFKB. A mutated Notch1 results in constantly activated NFKB, leading to uncontrolled growth and thus cancerous T-ALL cells. NFKB inhibitor PT is known to inhibit the NFKB pathway and induce apoptosis in cancer cells. It intervenes with NFKB, preventing the activation of cell growth genes. Researchers found that the coupled treatment of CJ and PT had a stronger impact on the T-ALL cells than either compound alone. As you can see from the figure, the combination of CJ and PT, represented by the bar with the hashed lines, result in significant increase of T-ALL cells with mutations in Notch1.